In this video, we present INGOT, an in silico network-driven combination therapy approach. In particular, we will demonstrate three features of INGOT using a signaling network related to triple-negative breast cancer. These features are importing the network, visualizing the network and predicting target combinations. First, we select the signaling network file. Note that for this demonstration, we use a human signaling network containing over 6,000 nodes that has been preloaded. We shall now visualize the human signaling network. First, we select the Visualize Preloaded Network option. In the configuration screen, we select the network and settings for visualization. For instance, triple-negative breast cancer view with 3 hops will visualize the network with molecules relevant to triple-negative breast cancer and those that are 3 hops away. The visualized graph either displays fold change or mutation frequency of the nodes. Fold change is used to measure changes in gene expression level obtained from the omnibus dataset. It is derived from the ratio of the gene expression intensity of the tumor sample to that of the normal sample. Mutation frequency reports the percentage of samples in the cosmic database that carry mutation in that particular gene. For fold changes, nodes are represented in green if they are overexpressed and in red if they are underexpressed. The node size indicates the extent of over or underexpression. The user can switch between from viewing fold change to mutation frequency by toggling the radio button. For mutation frequency, the size of the node indicates the frequency of the mutation. Larger sized nodes are mutated more frequently. Finally, to predict target combinations, click on the Action tab and select Compute Target Combinations. A screen will pop up and guide the user through the different steps. First, select the disease nodes related to the signaling network. Disease nodes are molecular species that are implicated in the disease, and regulating them may restore the physiological state. For example, SEMA5A, a pro-angiogenic gene, typically exhibits high expression level in triple-negative breast cancer. The disease nodes can either be selected from a list by the expert user or generated using rules. For instance, setting disease nodes as those having expression fold change more than or equals to 2.33 and with mutation frequency percentage more than or equals to 2 selects DST, SEMA5A, and CDKN2A as the disease node set. In the next step, we select the off-target rules. These rules are for computing an off-target score which is used to rank different target combinations. A lower off-target score implies a combination with potentially lower toxicity. In this video, INGOT seeks combinations that minimize perturbation of essential nodes and nodes with high weighted out degree. Nodes with high weighted out degree are more likely to propagate the perturbation. We then set the combination size as 3 and use the greedy approach with subgraph heuristic to compute the target combinations. This heuristic partitions the network into subgraphs, each containing one or more disease nodes, then for each subgraph, identifies suitable target to add to the combination. Target selection depends on the disease node cover and the off-target score of the resulting combination. Clicking on the Compute button initiates computation of target combinations. After computing the target combinations, the target combination nodes are highlighted in green and the disease nodes in red. The Disease Node List tab displays a summary of the disease nodes, their fold changes and mutation frequencies. A literature survey of these disease nodes reveal that they are implicated in metastasis and invasion, which is likely to be a high-priority cancer hallmark for triple-negative breast cancer. Finally, the Target Combination tab at the bottom left panel displays a summary of the best predicted target combination. This combination consists of SEMA5A, TP63 and SMAD3. Each of these components has been implicated in cancer by different research group. For instance, TP63, a P53 family member, controls a survival pathway that directly mediates cisplatin sensitivity in triple-negative breast cancer. Hence, the predicted combination is likely to be biologically relevant. Note, however, that further wet lab experiments are required to verify the predicted combination. In summary, INGOT uses a systems biology-based approach to predict target combinations and is useful in helping scientists analyze disease-related signaling network and to formulate hypotheses for guiding the design of further wet lab experiments.